Okay, I'm sorry there was a cutoff. Are you all with me now?
Yes.
Okay, I hope this does not happen again. Fine, we'll go back to where were we? Fine, so again, we will go back to what we were talking about. We were saying that enzymes are specific. Is this clear? That part is clear. Again, all the remaining pictures here will show you that you have an enzyme with a precise three-dimensional shape, and it has a depression known as the active site. No one will ask you what is an active site, but you need to understand what is it and know how to label it. It is the site that will fit with the substrate. And if the shapes of the active site and the substrate are complementary, they will fit together. If they are not complementary, they will not fit together, like this enzyme and this substrate. What is the substrate? The substrate is the molecule the enzyme is working on. Now, it's obvious here that the shapes of the active site and the substrate, are they complementary or not complementary?
Complementary.
Complementary. And that is why it will be able to work on the substrate and break it down to give you products. In case you want to know what is a product, the product is the final result of an or a reaction. What is produced after the reaction is finished. So going back to my enzymes. Okay, if you notice, I don't want you to memorize anything. I want you to understand. Understanding makes life very easy. So enzymes, mode of action, how enzymes work. What we just described is a hypothesis or a theory in biology known as the lock and the key theory. Okay? This is very easy to describe. If you get an essay question in paper 4 that asks you, how do the enzymes work? Or what is the lock and the key theory? You will simply say what we just said in the explanation, that the enzymes have a specific three-dimensional shape. And they have a specific active site. Okay, there is background noise, so I'm sorry, I'll mute all of you. If you want to, if you have a question, just raise your hand. Don't even know how to mute you. Okay, so if you have a question, raise your hand. So when we are asked, how do the enzymes work? You will say that they work by the lock and key theory. Lock and key theory, which is what we just described. You will mention in your answer that the enzymes have a precise three-dimensional shape, and they have a specific active site. This active site, a specific active site that also have a specific shape that is complementary to the shape of the substrate. So only one particular substrate with that specific shape will be able to fit with the enzyme. And no other substrate will be able to fit into the active site of the enzyme. This is the lock and key hypothesis. Did you understand what the lock and key hypothesis means? Anyone has any questions? Yes, Nadine. Wait, you are unable to unmute yourself? Yes, my dear. Yes, Nadine.
Miss, can you just repeat it again?
Of course, I'll repeat it again. We are talking about enzymes and how they work. Enzymes, you understand that they're made up of proteins?
Yeah.
Okay, you understand that they speed up reactions?
Yeah.
Okay, now what you need to know is that we have many different types of enzymes in your body, and we have an enzyme for every reaction. So enzymes are specific. So we don't have one enzyme in your body that works on everything. No, we have an enzyme for every reaction. Why? Because enzymes, they have a particular shape. They are described as having a precise three-dimensional shape. And in the body of the enzyme, we have a depression called the active site. And as you notice here, that the active site has a specific shape. Now, I will give you two substrates, substrate A and substrate B. This enzyme will bind with which substrate, A or B?
A.
A. Of course, A will be able to fit into the active site. Why? Because A is complementary in shape or not complementary in shape?
Complementary.
It's complementary in shape.
Complementary in shape. Anything hard about that?
No, thank you.
Welcome. This is called the lock and key hypothesis. Now, when the enzyme binds with the substrate, it will bind with the substrate temporarily for a short period of time. And when they are bound together, it forms something called the enzyme substrate complex. Why did I say that this complex is temporarily? Because will the substrate remain bound to the enzyme forever?
No.
No, it will eventually break to give you products, because this is actually what we want to do. We want to break down the substrate into products, like, for example, starch. We want to break it down into what? What is the smallest unit of starch?
Glucose.
Glucose and glucose. Okay, so another term to understand is when the enzyme binds with the substrate, just like what you will see now, it will form a complex. This is called the enzyme substrate complex, which is temporary. It doesn't stay forever because the enzyme will bind with the substrate, break it down into smaller pieces, and we will have the final what? Product. Okay, I'll go back to the very beginning so that no one gets lost. Everything we said until now, we only defined what is an enzyme, and we discussed how it works. We talked about the lock and key hypothesis or theory. Let's read it together, and if you have a question, stop me. Enzymes have a precise three-dimensional protein structure. Each enzyme has a dent or a depression into which the substrate can bind to, called the what? The active site. The active site is complementary in shape to the substrate, or in other words, when you see this, it means or the active site exactly fits into the substrate. That specific substrate is the only molecule that can fit perfectly into the active site. No other substrate can fit into the same active site. When the substrate moves into the active site, they become known as the enzyme substrate complex that is formed temporarily. After the reaction, the products, they will leave the enzyme's active site as they no longer fit, and the enzyme is now free to take up another substrate. What is a substrate and what is a product? A substrate is the material upon which an enzyme acts on, and the product is the substance produced at the end of the reaction. Any question in what we said? I read it with you word by word. Is everything clear? Okay, no one raised their hand. This is a picture here just showing you the lock and key mechanism. This is the enzyme, and this is the substrate. The substrate will fit here, but will not fit here because it's not complementary to this enzyme. It's only complementary to this enzyme. I hope now things are clear. Very easy. Any questions before I move on? Okay, now you need to study the names of a few enzymes. They're not a lot, maybe 3 or 4. And I think you heard about them. I'm sure you heard about the amylase enzyme. This amylase enzyme, what does it break down? What is its substrate?
Starch.
Starch is broken down by amylase to give us what?
Glucose.
Okay, this is what you took last year. You are not wrong. Amylase breaks down starch into another sugar called maltose. And then this maltose will be broken down by another enzyme that you'll take this year called maltase to give you what?
Glucose.
Glucose. And I'll give you an easy way to remember the name of the enzymes. They usually end in ASE, maltase, amylase, lipase, okay? Except protease. Protease, from the name, they break down what into what?
Polypeptides.
Proteins, amino acids.
Yes, you are both correct because polypeptides means proteins. So, 1 minute. Yeah, so the proteins protease break down proteins into amino acids. Found? Good. Now, there is a very important enzyme called the catalase. This catalase is a very important enzyme, and you will see it a lot in paper 6. Catalase is an enzyme found in your body, and it is also found in plants. This catalase breaks down a substrate known as hydrogen peroxide. Before you ask me, yes, you have to memorize it. Hydrogen peroxide is a substrate found in your body. and it is the result of a lot of chemical reactions that take place in your body. Hydrogen peroxide is very poisonous. It is very toxic. And if you want to know the chemical formula for hydrogen peroxide, it is H2O2. So hydrogen peroxide is very toxic. It is H2O2. It is the product of a lot of chemical reactions in your body. It cannot stay in your body because it will kill your living cells. So it will have to be broken down. It is broken down by an enzyme called catalase into two harmless products, which are water and oxygen. So please memorize this reaction. You need to only memorize the word equation for this reaction, which is hydrogen peroxide, which is very toxic and very poisonous, is broken down in your body by the catalase enzyme into two harmless products, which is water and oxygen. If you want the chemical formula, because some of you are curious, it's H2O2 broken down by the catalase enzyme to give me water and oxygen. And to balance that, you need to add a 2 here, a 2 here. Okay, H2O into water and oxygen. Is this balanced? It's not balanced now. 3, we'll balance it later. This is hydrogen peroxide to give us water and oxygen. Let me open a whiteboard and let's balance it together because a lot of you will want to know the chemical equation for that. Hydrogen peroxide is broken down in the presence of the catalase enzyme to give me water and oxygen. Now, is this balanced?
No.
No. So I'll put a 2 here and a 2 over there to balance it. Now you will have here 4 hydrogen and 4 oxygen and 4 hydrogen and 4 oxygen. This is a balanced equation. If you want to know the balanced equation, because a lot of you like to memorize the balanced equations. Okay, you're not obliged to know the balanced equation for that, just the word equation. This is hydrogen peroxide. Now again, why is hydrogen peroxide broken down? Why does it have to be broken down in your body?
Because it's toxic.
It's toxic. It's broken down into water and oxygen. So the name of the enzymes that you have to know, amylase. Any questions about amylase? Break down starch into
Maltose.
maltose. Very good. Now, who will break down maltose? Maltase.
Maltase.
Maltase. Maltase will break down maltose into
Glucose.
glucose. Excellent. Now we have protease that will break down what?
Protein.
Proteins into amino acids. When we study the lesson digestion, we will study 2 different types of protease because protease is a family of enzymes. Now you have another enzyme that breaks down fat. Who knows its name?
Lipase.
Lipase.
So the lipase enzyme breaks down fats into
Fatty acids and glycerol.
excellent fatty acid and glycerol. And you should know this because we took it in the lesson biological molecules. Now the new enzyme that you will study this year is the catalase. And you heard about it last year. Now the catalase breaks down what?
Hydrogen peroxide.
Very good. Hydrogen peroxide into
Water and oxygen.
excellent water and oxygen. If you want to balance that, I put a 2 here and a 2 here. Okay. Now, the remaining part of the lesson actually is mainly experiments that you need to understand. Let's go back to the notes so that I don't jump. So, catalase enzyme, let's read it together, found in all living cells such as liver cells and potato cells, because we always use potatoes in our experiments. Catalase breaks down the hydrogen peroxide into water and oxygen. Hydrogen peroxide is a waste product from many reactions taking place inside the body, and it must be broken down because it's very dangerous and it's very toxic. It kills your living cells. Most enzymes break large molecules, but some enzymes build large molecules from smaller ones. Any question? Okay. Any question? I don't want to move quickly. Is everything clear?
Can you give us an example for the building large molecules from smaller ones?
Very good. Very smart question. Now, if I will add amino acids together, my dear, what will I build?
Protein.
This is one example. working in the opposite direction. Okay.
Okay.
When you go to year 11, you will know that the reactions that break down large molecules are called catabolic reactions. I don't know if you took it in chemistry. And those that build bigger molecules from smaller ones, they're called anabolic reactions. If you know, fine. If you don't want to know it, it's okay. Okay? Now, we'll talk about enzymes and temperature. Enzymes and temperature. This is very, very, very important. And I'm going to jump directly to the graph because this graph is so important. You can't imagine how important it is. And what's nice about it is that you will not memorize, you will understand. Now, I want you to look closely at this graph. You have here temperature on the x-axis, and you have the rate of the reaction on the y-axis. Now, temperature is increasing on the x-axis, as you can see, and you have the rate of the reaction on the y-axis. What are we doing in this graph? We want to know how the temperature affects the enzyme. Now, I want you to talk to me about this part of the graph. Only that part of the graph. And tell me, what is happening to the rate of the reaction as the temperature is increasing?
Increasing as
It decreases.
What is decreases? What is decreasing? When the temperature increases, the enzyme will work faster or slower. What do you see from the graph?
Faster.
Someone said decreases. Who is this someone?